In the last objective, we talked about signal transmission in terms of signals that are synthesized, and we talked about how the cell get the signal to the target cell. In this objective, we explore reception. Reception is receiving that signal. All right. So now we're talking about the receptors. These are large proteins or large glycoproteins that are going to receive the signal. Now, when we say that, it is important to note that these receptors that are present, that are receiving the signal, these receptors are very specific about what signal that they will bind to. And so we focus on reception and it being highly specific in this particular objective. So first, let's explore what a receptor looks like, all right? So we know these receptors are large proteins or large glycoproteins, and we've talked about so far that these receptors are present on the target cell. Some of these receptors are embedded in the plasma membrane, all right? Some receptors are embedded in the plasma membrane. And so what we see here are these sort of Y-shaped receptors in this animation. The yellow illustrates the plasma membrane, all right? So your receptor has different domains or different parts, all right? So a domain is a part, and it has an extracellular domain. This is the part that sticks on or that's present on the outside of the plasma membrane or the extracellular portion of the plasma membrane. It has an intracellular domain, all right? So this is the part that extends through, okay, the plasma membrane into the cytoplasm, okay, into the cytoplasm. And it's also, so when we talk about the extracellular portion or extracellular domain, one of the characteristics of the extracellular domain is that it is hydrophilic. So if you would recall from the earlier chapters, hydrophilic means water loving. All right, so these parts have to be able to be specific for signaling molecules that are hydrophilic, okay, hydrophilic. You also have, in order for these receptors to span the membrane, part of this membrane must have a hydrophobic domain, okay, a hydrophobic part of the receptor, okay, so it can expand or extend through the plasma membrane. The intracellular portion of the receptor has to be hydrophilic because remember, it has to be able to be present in the cytoplasm inside of the cell. All right, so we have a hydrophilic domain that's located on the extracellular portion. We have a hydrophobic domain that extends, all right, that extends, and then we have a hydrophilic domain that extends into the cytoplasm. All right, now, in terms of what's going to bind to the hydrophilic domain located in the extracellular portion of the cell, we have what's called ligands, all right? So ligands. So ligands are the signaling molecules that are secreted by another cell, all right? Another cell. So these signaling molecules, ligands, are nothing but signaling molecules. These are going to bind to the receptor. But again, they have to be specific to the receptor, okay? They're not, all signaling molecules don't bind the same receptor. So these ligands are specific for this receptor, okay? And then what happens is, if you notice the difference between this image and this image is the intracellular portion of the receptor, okay? The intracellular portion of the receptor. So notice when the ligand binds the receptor, it triggers a conformational change. All right, it triggers a conformational change for the intracellular portion of the receptor. And so what this does is it allows for other proteins to bind, and eventually you're going to get that cascade of signaling events that we talked about in the previous learning objective. Other proteins are going to bind, signal to other proteins within the cell, and then eventually you'll have a biological response. Okay. So again, I gave you an overview of what's to come. So we'll talk about the signaling molecules. So the signaling molecules are going to fit the shape of the specific receptor, okay? So all signaling molecules will not bind the same receptor. These signaling molecules are specific for a certain receptor. Your receptor is going to have a specific shape that's specific for that ligand, okay? That ligand or that signaling molecule. So as stated on your slide, different cell types produce different receptors. A cell may synthesize many different kinds of receptors, which may change with the stages in its life cycle, okay? So all in all, the same signal can have different meanings for various targets. So we have highlighted here in terms of the receptors or the signaling molecules for the receptors, you have ligand, all right? So a ligand and a signaling molecule are the same thing. The ligand will bind to a specific receptor, okay? Now, your ligands can be either hydrophilic or hydrophobic. When the ligands are hydrophilic molecules, they will bind to receptors on the surface of the target cell. And that's what's shown here. You have a receptor that's embedded in the plasma membrane. So we know that from the last slide that this particular receptor has a hydrophilic domain that's located on the extracellular portion of the cell. So therefore, it can only bind hydrophilic molecules. So the only ligands that can bind to this receptor that's embedded in the plasma membrane, it has to be hydrophilic. Whereas there are some receptors that are located in the cytoplasm or that are located in the nucleus. In order to cross the plasma membrane, these ligands must be hydrophobic. Hydrophobic ligands will bind to receptors that are present in the cytoplasm or in the nucleus. Okay? Cells also have to, so we talked about the receptor and its specificity. So cells also have to regulate the reception. So yes, it can receive a signal, but sometimes the cell needs to amplify the effect of the signaling. They need to make the signal much, much greater. So maybe they need to make more of a particular molecule or that will eventually upregulate the cell's response. So to increase a cell's response, because remember, if we go back, remember we said that signals will bind to the receptors. The receptors will cause a cascade of signaling events, and that will lead to some biological effect. If I need to amplify the biological effect, the cell needs to regulate the number of receptors that are present, all right? So it's going to upregulate or increase the number of receptors that are present either on the membrane or within the cell, all right? So by increasing the number of receptors, more ligand can bind, or more signaling molecules can bind to the receptor to amplify the signaling effect. Okay, so to amplify that biological effect. So if you need more, the cells to divide more, if you need cells to grow more, if you need some, if you need to regulate some type of gene activity and you need more of it, then the cell will upregulate the receptors that are present. So you can increase the signaling that's happening inside of the cell and eventually the biological response. Now, what happens when I need to decrease some of the signaling? What happens if I have too much cell proliferation? What if, or cell division? So cell proliferation and cell division are the same thing. I use those interchangeably. So if I need more cell division and I need more growth, or I need more of some activity or biological response, again, if I need more of it, I'm going to upregulate it. But what if I need to slow it down? What if it needs to stop? Then I don't want to make the cell signal more. So to help with this, or to aid in stopping this process, we're going to down regulate the receptors. Okay, we're going to down regulate the receptor. So in order to down regulate, or to stop receiving this so many signals from so many ligands, we need to transport those receptors to lysosomes. We're going to destroy those receptors, okay? We're going to destroy those receptors and make them less sensitive to the ligands. Got it? All right. And so overall, we've identified mechanisms that make reception a highly specific process. We have said that receptors are highly specific in that they have a certain shape, and so therefore they are only able to bind to certain ligands. All right. And if we need, or if the concentration of something is low and we need to make more of it, then we know that the receptors can upregulate, bind, and then make more of whatever it is that it needs to make. If the concentration of something is high for the cell, then we know that we can downregulate the receptor by allowing lysosomes to destroy those receptors, and therefore we make less of something.